This is the last lecture on circulation and gas exchange. Gas exchange refers to the uptake of O2 from the environment, as well as the discharge of CO2 to the environment. So of course, our cells are generally, when they're doing aerobic metabolism, they need oxygen, and a metabolic byproduct of oxidative phosphorylation is CO2, and that has to be released back to the environment. And here is an axolotl. It's an albino axolotl, and you can see that it has gills. And we'll talk more about this animal a little bit later. Okay, one concept that we need to understand when looking at respiratory physiology is the idea of partial pressure. This is the idea that within a gas mixture, each gas individually exerts a pressure. So for example, in this picture here, we have gas A, which exerts a partial pressure, P sub A. We have gas B, which exerts a partial pressure, P sub B, and gas C, which exerts a partial pressure, P sub C. Now, recall that what pressure is. Pressure is force that's perpendicular to a surface. You could imagine, for example, this might be a box, and so these molecules of gas are colliding with the wall of the box and exerting a force on the wall of the box. And so pressure is defined as force divided by the surface area that the force is acting on. So, for example, when you fill up a tire, the units are pounds per square inch. So pounds is a unit of force, and square inch would refer to the surface area that the force is acting on. Okay, so each of these pressures, individual pressures, can be added together. So for example, if we had all three of these gases in another box, well, the total pressure, P total, is simply equal to this sum of P sub A plus P sub B plus P sub C. You just simply add them together. Let's now use the concept of partial pressure to do some calculations. Given that we know the total atmospheric pressure is 760 millimeters of mercury, and that air is composed of 21% oxygen, what is the partial pressure of O2, or P sub O2? Okay, to solve this problem, we'll write things out. So we have 760 millimeters of mercury. That's our total pressure. Now we know that air makes up 0.21 of this, so we write that out, 0.21. So we simply multiply those two together and we'll get 160 millimeters of mercury as our answer. Okay, so at sea level, given that air is composed of about 20% oxygen, the partial pressure of oxygen is 160 millimeters of mercury. So partial pressure gradients are important for understanding gas exchange. One situation that arises quite often in biology is we'll have an interface between gas and a liquid. So for example, in your lungs, we'll talk about alveoli, which are little sacs where gas exchange occurs. And so there's air in the alveoli, so that's referred to as the alveolar airspace. And oxygen from that alveolar airspace will diffuse into liquid that lines the inside of these alveoli, which are these little sacs. And from there, it'll enter through the epithelial layer of the alveolar sac, and then into interstitial fluid, and then into pulmonary capillaries. So you basically have an interface there between gas, which is the alveolar airspace, and then liquid, starting with that liquid which lines the alveoli. So this is one common example in which we need to understand what would happen at such an interface. So let's look at this picture here. At equilibrium, I'm showing you a picture in which you have oxygen molecules shown in green, and that's in the gas within the air within, say, the alveolar airspace. And then you have some oxygen that's diffused into the liquid within, say, the alveolar, each alveolus. And you'll notice here that there's quite a difference in the number of molecules. There's about 30 times more of these green molecules in the gas phase than in the liquid phase. And that's meant to represent the idea that oxygen is much less soluble in water than in air. So at equilibrium, this is the situation you'd have. So let's imagine for a second, it's actually going to be lower than this, but let's imagine we have a PO2 of 160 millimeters of mercury. In the actual alveolar airspace, it's closer to about 100. Well, let's just take a situation in which we know that the gas has a PO2 of 160 millimeters of mercury, which is the same value as what we'd found in air at sea level. Well, what would be the PO2 in the liquid? It turns out that the way PO2 in the liquid is defined, it's actually just defined under these equilibrium conditions. In other words, if the gas is in equilibrium with the liquid, meaning that for every molecule that enters into the liquid from the gas, there's a molecule that moves back from the liquid to the gas. So there's no net movement of oxygen. So that's an equilibrium condition. So under these conditions, if the PO2 is 160 millimeters of mercury in the gas, then it's also 160 millimeters of mercury in the liquid. So we can write that down here. It's 160 millimeters of mercury, given that we're at equilibrium. Okay, given, this is a table from your book, and it just shows you some of the values we've been talking about. So for example, in air, the O2 partial pressure is 160 millimeters of mercury. In water, that's in equilibrium with air at sea level, it would be identical. It'd also be 160 millimeters of mercury. So the ratio of partial pressure is 1 to 1, okay? Now, if we look at actual O2 concentration, it's very different. In air, it's 210 millimeters of oxygen per liter of gas. So that makes sense, right? 21%. 210 divided by 1,000 is 21%. In water, the value is much lower. It's about 7 milliliters of oxygen per liter of water. And so now if you take the ratio of the 210 to 7, that's a ratio of about 30 to 1. And that was what we represented in the earlier diagram, showing that there's at an equilibrium condition, there's far more oxygen molecules in the air compared to in the water. A couple other things to note. Air has a very low density, which is what you'd expect, but then water has a very high density of 1 kilogram per liter. So water is essentially 770 times more dense than air. They also differ in viscosity. So air has a viscosity of 0.02 centipoise, and water has a viscosity of 1 centipoise. And so this is a ratio of 1 to 50. So in other words, water is 50 times air. So due to these differences between air and water, there are very large differences in the way animals will breathe, depending on whether they're breathing air or water. Breathing air is relatively easy, and it doesn't have to be particularly efficient. So in fact, the way we breathe, the way mammals breathe, is actually fairly inefficient. In a given volume, there's less water O2 available in water than in air. And so for an animal such as a fish or any sort of animal living underwater, that leads to some difficulties for obtaining oxygen. So obtaining oxygen from water requires greater efficiency than air breathing. And aquatic animals such as fishes and lobster must expend a considerable amount of energy to obtain the oxygen that they need. Okay, whether you're breathing air or water, the exchange of gas occurs across what's called a respiratory surface. And the exchange occurs solely by diffusion. Respiratory surfaces vary by animal and can include skin, gills, trachea in insects, and lungs. So here's a very simplified diagram of a respiratory surface. So we're showing, for example, here a square animal or a rectangular animal. It has a respiratory surface shown in blue here, and then I'm showing a simple sort of circulatory system of some sort. And so the idea is that oxygen, for example, will have to enter through the respiratory surface by diffusion. It'll then be picked up by the circulatory system and then moved around the animal. So this the rate of diffusion across the respiratory surface is governed by a rule that was worked out by Fick and is sometimes referred to as Fick's laws of diffusion. The rate of diffusion, that is the number of molecules per unit time, is proportional to the surface area of the respiratory surface, the concentration difference. So this would refer to the difference in oxygen partial pressure, say in the medium, and the partial pressure of oxygen within the animal. And then this is then divided by the thickness of the membrane. So the membrane refers to this, it's not like cell membrane, but rather the whole surface through which the oxygen would have to diffuse. So for example, respiratory surface might include a layer of water on the surface. Then the cells, for example, in the lung, you'd have epithelial cells that make up the alveoli, which are these little sacs in the lungs. Then through the interstitial fluid, and then through the endothelium of a pulmonary capillary. So that whole thing would essentially be the diffusion path, and that would be the thickness of the membrane that we're talking about here. So let's think about what would happen. For example, if the surface area or concentration difference is doubled, what will happen to the rate of diffusion? Well, if you increase the surface area by 2 times, then the rate of diffusion will be proportionally higher, so it'd be twice. If you increase the concentration difference by a factor of 2, then the rate of diffusion would also increase by a factor of 2. So there's just a linear dependence there. Furthermore, like if the thickness of the exchange membrane is halved, what will happen to the rate of diffusion? Well, so if we could make the membrane thinner, then that'll actually increase the rate of diffusion. If we make conversely, if we make the thickness of the membrane greater, then that'll decrease the rate of diffusion. Say we double the thickness of the membrane, then the rate of diffusion would be halved. So these rules are fairly simple, but they help us understand why respiratory systems and surfaces are designed the way they are. For example, in your lung, you'll have a very large surface area. In order to maintain the concentration difference between the medium and the inside of the animal, you're going to need to do ventilation, which means you're going to need to renew the medium above this respiratory surface. So, for example, bringing fresh air into the alveolar airspace. And then the thickness of the membrane is very important. So if you look at your lungs, they're actually very delicate structures. The alveoli are these very tiny sacs. They don't have any cilia in them. They're just a very simple epithelial layer, very thin. There's a very, very small amount of interstitial fluid, which is the fluid between cells. And then you get to the pulmonary capillaries, which are very closely associated with the alveoli. And so everything in your body would tend to maximize this rate of diffusion because you want to get oxygen into your body so you can do oxidative phosphorylation at the levels of cells in your body. Okay, many animals have gills, so particularly aquatic animals. So gills are outfoldings of the body that create a large surface area for gas exchange. So for example, Let's take this very simple animal. It's our box with the simple respiratory surface here. Well, how can we increase the surface area? Well, one simple way to do it is to have an outfolding or outpocketing, and that's what gills are essentially. Developmentally, they're outpocketings of a respiratory surface. And you can see the surface area here is about 3 times larger, roughly, than what you see here. In addition to this, you want to have some ventilation. So ventilation is a very general term. It refers to the movement of the respiratory medium. So for example, water that would contain oxygen in, say, a pond, over the respiratory surface. So again, we can think about our axolotl. It has gills, and it lives in an aquatic environment in lakes in Mexico. And so if there's a bit of a water current, say in the lake or river, that would be good. It would bring fresh oxygenated water to the gills of the axolotl. Now, if it turns out that the axolotl is in a space in which there isn't a lot of water movement, well, the axolotl actually has muscles at the base of its gills, and so it can actually move its gills back and forth, and that's another way of renewing the medium surrounding its gills. And why would you want to do that? Well, if you didn't have any movement of the medium over the respiratory surface, then locally you would use up all the oxygen locally, right? You just have sort of stale water in a sense used up with the lower partial pressure of oxygen. By bringing in fresh water, you're able to bring in water that has a higher partial pressure of oxygen, and that maximizes this gradient we talked about, the gradient of partial pressure between the outside and the inside of the animal, and therefore that increases the rate of diffusion of oxygen or other molecules into the animal. This figure from your book illustrates some of the different types of gills that exist in aquatic animals. In marine worms, you have structures called parapodia. These function as gills. They also function, they're useful for crawling and swimming. Crayfish have gills. They're within the exoskeleton. And crayfish actually have an efficient system for pumping water over their gills. Finally, starfish have gills that are essentially these tubes that are continuous with the coelom within the animal. And so as the animal refreshes the water inside the coelom, then there can be exchange of oxygen between this fluid in the coelom and the interior of the body of the animal. So previously we talked about the difficulty of breathing water. Water has a lower, for a given partial pressure, it has a lower amount of oxygen per volume. So there's a lower concentration of oxygen. It also is much heavier, much denser than air, so that presents difficulty. Moving the medium over the respiratory surface is going to be more difficult. And water is also more viscous than air. So viscosity is really primarily a problem if you're trying to move something back and forth. For example, the way we breathe air in, it then stops, and then we breathe it out. So we're sort of moving it back and forth. Now that's very difficult with a very viscous medium. So for these reasons, fish gills use a unidirectional flow system where the medium always flows over the gills in one direction. So you don't have this problem of the medium going back and forth. So for example, water will flow into the mouth of the fish. It'll then flow over the gill arches, and the gills are protected by the operculum, which is kind of a bony plate. And then there's a gill slit, and then the water will eventually end up exiting the gill slit. So water's always going unidirectionally through the fish. It never goes back out the mouth, because that would be very metabolically costly. It's to take something as dense and viscous as water and to breathe it in, and then to push it back out. Although it could be done, it'd be very, very tiring, very difficult. So fish gills are also highly efficient. More than 80% of the oxygen dissolved in the water is removed as the water passes over the respiratory surface. So they have a very nice system of unidirectional flow. They're also very efficient at extracting oxygen. And there are essentially two ways that fish can breathe. They may actively breathe with coordinated movements of the mouth and the gill covers. So it's kind of a pumping action that you'll do. So if you go to an aquarium and you look at fish, you can sometimes see them doing this. Additionally, some fish can also simply open their mouth, and as they're moving forward, the water will flow in and then across the gills and out through the gill slit. And so some fish are actually quite good at doing both. They'll do both, the pumping motion with the mouth and the operculum, and then they can also use this other form of ventilation, which is referred to as ram ventilation. Now, it turns out there are some fish, so for example, some species of shark that will exclusively use ram ventilation. These tend to be the most active sharks, the ones that are sort of very fast. They've actually lost the ability to do the pumping motion with the mouth and the operculum, and so they rely on simply moving forward at all times. So they ventilate their gills by swimming with their mouth open. And if they get caught in a net, say, then they'll often die. These will be the sorts of fish that die because they're not able to continue to move forward all the time. Okay, so again, we talked about the unidirectional flow. You have it within the fish under the operculum. You have a number of gill arches. On each gill arch, you have essentially a series of gill filaments. And then if you zoom in on the gill filaments, you'll see these things called lamella, singular, or lamellae, plural. So these are plate-like structures, and within these structures you have, they're quite thin, and then they have capillaries. And so you can see oxygen-poor blood coming in, going through the capillary. It picks up oxygen as the water goes by it, that has the oxygen. The water, as it goes by, it loses oxygen to the capillaries, and so it becomes deoxygenated, at least partially. And then this blood that has now picked up the oxygen is then goes back to the body of the fish. And so this turns out to be an extremely efficient system. We're going to skip over the tracheal system of breathing in insects. Okay, we've talked about gills, which are mostly, most prevalent in aquatic animals. Well, lungs are another way of breathing. So gills, remember, are an outpocketing or outfolding. Lungs are the opposite. They're an infolding of a body surface. So again, let's take our very simple animal, which is this rectangular animal with a respiratory surface. In order to increase that surface area, another way of doing it, instead of an outpocketing, you can have an infolding. So that's essentially what a lung is. And of course, it could be as simple as that, a simple pocket. And you'll actually see lungs as simple as that in certain land snails. But in animals that are more metabolically active and complex, such as us, this simple inpocketing becomes very, very elaborate into a sort of tree-like structure, which is our lungs. Circulatory system can be either open or closed. So recall, we talked about this earlier. So an open circulatory system would be found in many arthropods, such as crayfish, lobsters, spiders. In an open circulatory system, the blood is not, maybe in blood vessels initially as it leaves the heart, but then the vascular system actually opens up, and the hemolymph is then able to simply mix with the cells and then is transported back to the heart. A closed circulatory system is what we have. Our red blood cells, for example, never ever leave our cardiovascular system. Other animals that have closed circulatory systems are things like annelid worms, and certain mollusks like the cephalopods, such as squid and octopuses, which are very metabolically active. They also have a closed circulatory system. So basically, we have to have a combination of some sort of respiratory surface, and this is then paired with a circulatory system that is able to then distribute the gas around the body. In terms of lungs, there's actually quite a lot of variety in terms of structure and so forth. The size and complexity of lungs correlates with an animal's metabolic rate, for example. Also, there's correlations with the lifestyle of an animal. For example, in amphibians, a lot of gas exchange can actually occur across their moist skin. So, for example, a frog has very moist skin. So oxygen can diffuse in, carbon dioxide can diffuse out directly through the skin. They also have moist mucous membranes within the mouth that also allow for gas exchange. And finally, frogs do have a relatively simple lung, which allows for gas exchange. So when frogs are underwater, of course, they're going to exclusively use the breathing across the skin. When they're on land, they then they can continue to breathe across the skin as long as the skin is moist. And they'll also breathe using their lungs, as well as there'll be some gas exchange within the oral cavity itself. Okay, well now look at our own body, which is of course a bit more complex than a frog's in terms of its respiratory system. We have a system of branching ducts that convey air to the lungs. Air is inhaled through the nostrils. It's filtered by hairs within the nostrils. It's warmed and humidified. So by the time the air actually reaches the alveolar air space within our lungs, it has a relative humidity of 100%. In other words, it couldn't be any more humid at the internal temperature of 37 degrees centigrade. So air enters. You have something called a pharynx. So this is essentially a common tube that leads to both the trachea as well as the esophagus. The esophagus is behind the trachea. And of course, the problem is there is a little bit of a design problem here, which is we want to breathe and also ingest food through the same tube, through the pharynx. And so we need to be able to direct things in the proper direction. So when we're breathing, the trachea is open and the esophageal sphincter is contracted. Okay, that's good. So we're able to breathe. Now, if we have a bolus of food, we're going to have to redirect that bolus to the esophagus, and we don't want it to go down the trachea. So in order to achieve this, when you swallow, that action moves the larynx upward. So the larynx is the voice box. It's shown here. It actually moves upward, and the epiglottis tips over from being in an upward position to down, and it interacts with the glottis, which is up, and this prevents food from going down the trachea or windpipe, and it will go down the esophagus. Okay, when we're breathing, air passes through the pharynx, then it goes through the larynx, which is also known as the voice box. That, of course, contains the vocal cords. It then moves on to the trachea. So notice the larynx actually has these rings of cartilage. So if you feel in your neck, you can feel the larynx and you can feel those rings of cartilage. And then this idea of rings continues down with the esophagus. So these rings are very similar to, if you look at a tube for a vacuum, the rings allow for flexibility. So the larynx and the trachea are actually quite flexible tubes, but at the same time, they won't collapse. They're very strong. And so that's exactly how a tube for a vacuum cleaner works. Okay, so you have rings of cartilage. You then go to the bronchi, which are the major branches. And then eventually you end up with much smaller branches called bronchioles. And from the bronchioles, that leads then to the alveoli, which are the small sacs where the actual gas exchange occurs. So exhaled air passes over vocal cords and the larynx to create sound. So that's how we talk. So when we're speaking, we're essentially exhaling. And then in between those exhales, we inhale, and then we continue to exhale as we're talking. So one of the problems, of course, is there's a lot of surface area within the respiratory system. And so this allows for pathogens such as bacteria and viruses to attack us. So how do we prevent that? Well, most of the respiratory surfaces have cilia, and they also have, so there's ciliated cells, and there are also mucus cells, cells that secrete mucus. And these are the epithelial of the air ducts. And what these cilia do is essentially will move the particles that you breathe in by accident or that are in the air, dust, viruses, bacteria, things like that, and they move them up to the pharynx. So this is referred to as a mucus escalator. The mucus is very effective at picking up the bacteria and dust and foreign particles. It essentially continuously cleans the respiratory system and allows these particles to be swallowed at the level of the esophagus. Okay, the actual gas exchange will take place exclusively at the alveoli. These are air sacs at the tips of the bronchioles. Oxygen diffuses within each alveoli. There's a moist film on top of the epithelial layer of these alveoli, and so oxygen. So that's a condition. You wouldn't be able to pick up oxygen unless you had this moistness, because the oxygen initially diffuses into the moist film, and then through the actual epithelial layer of the alveoli, from there into the interstitial fluid, and then into the pulmonary capillaries. Carbon dioxide diffuses from the capillaries across the epithelium and into the airspace, so it simply goes in the opposite direction. So here we can see a picture. We've taken a little section of the lung and expanded it. We can see, for example, here, terminal bronchioles, and then that leads to these things that look kind of like grapes. These are the alveoli. They're organized in bunches. And we can see, we've talked previously about the circulatory system. For example, we can see a branch of the pulmonary artery. So this is, again, defined as, an artery is defined as a blood vessel that leads away from the heart. So it has oxygen poor blood, and at the same time, these can, so then we have the pulmonary capillaries. These will pick up oxygen. So this is shown as purple, so they're picking up oxygen. And then finally, they have a fairly high level of oxygenation, and that's shown in red here. So the pulmonary vein here is then heading back towards the heart, and it has a higher level of oxygen. So it's going to go to the left atrium, and from there to the left ventricle, and then out the aorta. This is a close-up of the same diagram. I wanted to show you this because remember when we talked about the rate of diffusion? So the number of molecules that diffuse per unit time. Well, recall that that's dependent on things like the surface area. So if we look at this, having these alveoli, these sacs, well, that tremendously increases the surface area, okay? Another thing that increased the rate of diffusion was the concentration difference. And we'll talk about, instead of concentration, we'll talk about the partial pressure of oxygen. Well, within the alveolar airspace, you have a certain partial pressure of oxygen. And then the relevant partial pressure would be the partial pressure within the pulmonary capillaries. And so basically your body is designed to maintain a certain level of partial pressure within the alveolar airspace, and you do that through ventilation by breathing in and out. You're constantly bringing in some new fresh air to mix with the older air that's already in the lungs, and that maintains a fairly high partial pressure of oxygen, although it's a bit lower than the actual partial pressure of oxygen in the atmosphere. But you do that by ventilating. At the same time, you have the circulatory system that's bringing blood through, and so it's bringing in essentially blood that's deoxygenated. So again, that helps to increase the concentration difference, and that increases the rate of diffusion. And then the final thing, remember, in the denominator of that equation was the thickness of the membrane. Now recall, we're not talking about a plasma membrane, but we're talking about a membrane that's consists of various layers. So within the alveoli, you have this moistness, this layer of water. The oxygen diffuses into it, then diffuses into through the actual cell membrane and through the cells of the epithelial cells that make up the alveoli. There's a very small amount of interstitial fluid, very, very little space, because you can see here we're trying to minimize this distance of the thickness of this membrane. And so the pulmonary capillaries are very closely associated with the alveoli. And so that also speeds up the rate of diffusion. And the capillaries also themselves are very simple. They're just a single epithelial layer called endothelium. And so again, that maximizes the ability of your body to pick up oxygen by keeping that membrane through which the oxygen has to diffuse very thin. As I've mentioned a number of times, the alveoli have to have a thin layer of fluid lining them. If they didn't, you wouldn't be able to pick up oxygen effectively. Now, this turns out to be a little bit of a problem because having this layer of water inside each alveolus tends to collapse the alveoli. So for example, you probably are familiar with the idea of surface tension. So surface tension is sort of like a skin-like phenomenon in which the water molecules are attracted to each other because they're polar. Recall that oxygen is electronegative, and so it will tend to attract electrons, whereas the hydrogen ends of a water molecule tend to have a partial positive charge, because they've essentially unequally sharing their electrons with the oxygen. And so water is polar, and because of that, it tends to sort of stick together. So it'll chain together. And due to that, it has a tendency to collapse. And if you had, say, a spherical situation like this, like the alveolus, well, there'll be a tendency for the water to collapse together into a spherical ball of fluid. And of course, you don't want that, right? Because then you wouldn't be able to breathe. You want to maintain an open alveolus. And so that's actually problematic. And we'll talk about this a little more in the next slide. So to prevent the collapse of the alveoli, you have secretions called surfactants. You have special cells within the alveoli that release these surface active agents called surfactants. These coat that inner surface, and what they do is they reduce the surface tension of water. So they make the molecules of water, they essentially make them interfere with the attraction between the water molecules. The surfactants in your body are a mixture of phospholipids and proteins. And so because of these surfactants, your alveoli don't collapse and you're able to breathe, which is a good thing. Now, it turns out that preterm babies lack surfactant, and so they're vulnerable to what's called respiratory distress syndrome, or RDS. So we're not talking just about a couple weeks preterm, but about if the babies are about 6 weeks preterm, then they don't have surfactant. So back in the 1950s, RDS killed about 10,000 infants each year in the United States because there simply wasn't any treatment for them. They had difficulty breathing and they would die. But then, scientists figured out that if you could apply artificial surfactants, then that would allow these babies to breathe and they would live and actually thrive. Okay, we mentioned the definition of ventilation. So ventilation is essentially movement of the medium that contains the molecule of interest, such as oxygen, over the respiratory surface. And there are different ways of doing that. We talked about how ventilation occurs in, for example, fish. So we also ventilate. And so the way we do it is we alternate inhalation and exhalation of air. We're going to skip 2 sections. how an amphibian breathes, and we're also skipping how a bird breathes. Okay, mammals breathe by ventilating the lungs using what's often referred to as negative pressure breathing. So how does this work? Well, when you inhale, what you do is you activate skeletal muscle. So the diaphragm is this cone-shaped sheet of skeletal muscle. And when you contract the skeletal muscle, the diaphragm flattens out. And so that tends to increase the volume of the lungs. At the same time, and actually you can test this out yourself, if you put your hands on your chest and you breathe in, you'll feel that the rib cage actually moves outward and upward at the same time. And that again also increases the volume of your lungs. And so when you increase the volume, you will decrease the pressure within the lungs relative to the atmospheric pressure. And so the air will have a higher pressure in the atmosphere compared to the pressure within your lungs. And air will simply move down a gradient of pressure from higher pressure to lower pressure, okay? And then essentially the opposite phenomena occurs when you breathe out. When you breathe out, you relax, and the diaphragm moves, as it relaxes, it lengthens, and it actually goes back to being cone-shaped. And again, you could do this on yourself. You put your hands on your chest, breathe in, and then breathe out. You'll find that your rib cage goes backwards and downwards. And so that combined with the diaphragm moving upward means that the volume occupied by the lungs decreases. And as you do that, that essentially squeezes the lungs and increases the pressure. So now the pressure in the lungs is higher than that of the atmospheric pressure, and the air will move down its pressure gradient from being an area of higher pressure within the lungs to an area of lower pressure in the atmosphere. So the way we breathe is referred to as tidal breathing. We breathe in and out just the way a tide would move in and out on a beach on the shore. So we have a volume called tidal volume. This is the volume of air that's inhaled with each breath. And we can actually clinically look at some of these things. For example, one interesting thing to look at is called vital capacity. So if you ask someone to breathe out and then breathe in, as strongly as they can, then that would be this maximal tidal volume, and that would tell you something about the health of the person. So as people are young and healthy, they'll tend to have a higher vital capacity. After we exhale, there's always a bit of air left over in the lungs, and this is referred to as the residual volume. And there's really no way, no matter how tightly you squeeze your rib cage and so forth, you can't get the last remaining bit of air out of your lungs. As we age, this residual volume tends to increase. It's just simply harder and harder to get the last bit of air out of the lungs. And this has to do with changes in strength of muscles, but also primarily has to do with changes in the properties of the lung itself. The lungs essentially become less able to change volume, a little stiffer as you get older. Now, of course, that can be reversed by exercise and healthy diet and so forth. That can maintain healthy lungs. But if you have a higher residual volume, then the vital capacity will go down. And so again, that's something you could test if you were trying to look at the health of a person's lungs. So within each inhalation, you essentially are bringing in fresh air from the environment. So that's going to have about 21% oxygen, as we mentioned earlier. But that's going to mix with the air that's already in the alveolar airspace and within the bronchioles and so forth. And that air is oxygen depleted, not completely depleted of oxygen, but it certainly has less oxygen than in the atmosphere. And so the actual air that reaches the alveolar airspace, the space within those alveolar sacs, is never perfectly fresh. It'll have It'll always be a mixture of the oxygen depleted residual air as well as the new air that's coming in. Therefore, the PO2 in the alveolar airspace is less than the PO2 of air. Okay, our final topic for this lecture is the control of breathing. So as I mentioned, the diaphragm is skeletal muscle, and the muscles associated with the ribs are also skeletal muscle. And so it's not surprising. Remember, we have voluntary control over most of the skeletal muscles in our body. And so it's not really all that surprising. We can essentially command the breathing circuits, which are located in the brainstem, to stop breathing, or you could essentially command them to breathe faster or slower. So for example, here's a child who's voluntarily holding his breath. But most of the time, breathing is largely regulated by involuntary mechanisms. In other words, you really wouldn't want to spend your whole day thinking breathe in, breathe out, breathe in, breathe out. That would be sort of a waste of mental energy. So most of that's taken care of by mechanisms within the brainstem that simply do this for you. So the breathing control centers are found in the medulla oblongata, which is of the brain, which is the lower brainstem. The medulla regulates the rate and depth of breathing in response to pH changes in the cerebral spinal fluid. We'll talk more about that in the next slide. So, as I mentioned, most of the time we're generating ATP through oxidative phosphorylation. So, as the name implies, that's an aerobic means of generating ATP, and it requires that we breathe in oxygen. At the same time, we're generating CO2. So that CO2 is essentially a metabolic waste generated by all the cells in our body. So it turns out that for controlling breathing under normal conditions, when you say at rest, your body mainly pays attention to CO2, okay? And also pH. So CO2 levels and pH are intimately related. As you increase CO2 levels in your blood, the pH will decrease, for example. So our normal blood pH is about 7.4. If we, for some reason, were to get out of balance with our breathing, imagine that we let the levels of CO2 build up in our tissues. We could do this by voluntarily, for example, holding our breath. Well, the CO2 levels would increase, right? Because we wouldn't be exhaling CO2. pH of our blood would fall also at the same time. And so our body would then detect that. So there's a couple different routes through which our body detects that. The medulla, which is the lower part of the brainstem, detects a decrease in pH of the cerebral spinal fluid. So the cerebral spinal fluid is found in the ventricles and also in the central canal of the spinal cord, and also surrounding the spinal cord under this sheath that surrounds the spinal cord. So you might ask, well, how is it possible that there's a change in the pH of the cerebral spinal fluid? Well, it turns out that CO2 as a gas can move through cell membranes quite easily. So if the CO2 level were to rise, it can actually traverse the sheath that surrounds this spinal cord and so forth in the brain, and into the cerebral spinal fluid and have an influence on pH. So the medulla oblongata essentially pays attention to pH, but pH is really indicative of change in CO2. Now you also have, so this would be central chemoreception, essentially chemoreception by the brain. You also have peripheral chemoreception, and that's carried out in the carotid arteries in organs called the carotid bodies, and in the aorta, you have the aortic bodies. So these are sensors in these major blood vessels that can detect decreases in pH also. They can actually also detect CO2 and oxygen. So let's imagine that the pH falls and there's an increase in CO2. Well, that should indicate that we're not breathing enough. So these carotid bodies and the aortic body would be stimulated. The receptor cells there would be stimulated. They would send a signal to the medulla that would say essentially, you should breathe faster and deeper. The CO2 diffuses into essentially the cerebrospinal fluid of the medulla oblongata, leading to a pH change. A decrease in pH stimulates the medulla oblongata. So it itself also detects this change and says, hey, we need to breathe a bit faster and a bit deeper. So then those signals are then sent out along through neurons called motor neurons to the diaphragm and to the muscles of the rib cage. And so you'll then breathe faster and deeper. This will then allow you to essentially offload or get rid of that extra CO2 that had built up in your blood. And as you do that, the pH will naturally rise, and that'll bring the blood pH back to 7.4. Okay, to recap a little bit of what I just mentioned, because I know it was a lot, sensors in the aorta and carotid arteries monitor oxygen, CO2, concentrations in the blood. They also can monitor pH, as shown here. The medulla will monitor primarily changes in pH. So all these signals, signal the breathing centers, which respond as needed, as we just mentioned earlier. Now, it turns out we also have, as mentioned here, we have oxygen sensors in the carotid arteries and in the aorta. And we also have central oxygen sensors within the medulla oblongata, which your book doesn't mention, but they're there also. So why would we have these? Well, most of the time during our normal day, our body really just pays attention to these CO2 levels. And the pH sort of corresponds to the CO2 levels. So those two things go together most of the time. Now, there are situations in which your body really needs to pay attention to oxygen. So, for example, if you were to go to a very high altitude, then the oxygen level in the atmosphere would be low enough that these oxygen receptors would essentially become activated, and that would tell your body to breathe more, faster, and deeper. And there may be other situations, such as a disease state or something like that, in which oxygen levels may actually fall within the blood. And again, at that point, these oxygen sensors would become very important. So basically, it's sort of like a backup system. There are two systems. The main system is most of the time our body's paying attention to CO2 levels, but there are situations in which it makes sense to also pay attention to oxygen. And so you have those oxygen sensors also. And that pretty much brings us to the end of control of respiration.